Hello everyone, thank you for joining today's webinar. It's my pleasure to welcome you to the Materials Science section as part of Waters Asia Pacific LC Symposium 2021 for accelerating materials innovation with modern separation technologies. My name is Jen. I'm the System Marketing Manager for Waters LC and the MS products in Asia Pacific, and I will be the host for today's event. For today's event, we will begin with my colleague Charlotte's presentation to introduce Waters' liquid chromatography portfolio. Then we will have four featured topics from different speakers to talk about the needs, the challenges, and adapted technology in the different application areas such as fine chemicals and polymers. Before we begin, I would like to draw your attention to one housekeeping item. On your screen, you can see the main presentation screen. If you hover your cursor to the CC button, you may select the appropriate language subtitle according to your preference. And please take a few minutes to complete the survey at the end of today's live event. Now, I would like to pass over to the first speaker for today, Charlotte, who will introduce the current Waters liquid chromatography portfolio.
Hello, and thank you for joining us today. My name is Charlotte Yu, and I am a Separation Business Development Manager at Waters Asia Pacific. As many of you here already know, Waters has launched several new products in recent years with innovative technology. To start today's session of the LC Symposium, I'd like to start by giving you a short overview of the current Waters LC portfolio. As a scientist, we have questions every day. Do my samples contain toxic substances? Is our product safe? Is my data accurate? Our goal is to help you, our customers, confidently make critical decisions to move your business forward. From fundamental research to method development to routine testing, our integrated, robust offering of instruments, chemistries, informatics, and support addresses your research complexity. Our chromatography portfolio provides you with abilities. First one is usability. Our platforms support a consistent user experience, whether you are using an LC with optical detection for routine analysis or an LC with mass spectrometry for trace level analysis. Scalability. Our portfolio allows you to scale to meet your needs regardless of chromatography platform, column, or solvent. For many applications, LC configurations are paired with consumables and informatics, minimizing the time being spent from sample to result. Transferability. Our long-standing LC methods will run from instrument to instrument and lab to lab, producing the exact same data. As you develop or transfer your methods, you can rely on Waters to keep them fit for purpose as they move from development to routine use. Dependability. Our application scientists and global technical support teams answer your questions, help develop methods, and problem solve every step of the way. We are here to maximize your research and the innovations that result. There are many examples of the innovations in LC that we have brought to market in our history as a leader in separation science. The Alliance HPLC has been the backbone of many labs for over 20 years. But there may be no better example of how Waters has enabled scientists to gain competitive advantage than the advent of the Acuity UPLC in 2004. Since 2004, each addition to the Acuity family has been designed to allow a broader range of scientists to benefit from this game-changing family of products without compromise what they do. And with this industry following Waters' lead, the face of chromatography has changed. We have also added new specialty separations technologies such as convergence chromatography with UPS squared, polymer characterization with the ACQUITY APC system, and process monitoring with Petrol. Finally, we introduced several products in recent years to meet customers' needs and to solve another challenge. The Waters portfolio can be broken down in many different ways. So I would like to introduce our portfolio in 5 overlapping groups. The first one is analytical LC systems. These systems allow you to conduct your routine analysis with confidence. This group includes the original ACQUITY UPLC, the H-Class Plus, the Achilles PLC, and the ACQUITY Premier. These products provide the flexibility for easy method development, reliable method transfer, or uncomplicated routine analysis. Next one is LC for MS. High-performance LC technology delivered in optimized configurations that power up the sensitivity, reproducibility, and robustness of your MS analysis. Here I would include systems such as the ACQUITY I-Class Plus, the ACQUITY M-Class, and new ACQUITY Premier system. Next, we have system solutions. These fit-for-purpose solutions, like the Petrol system or the ACQUITY APC system, were designed to address application-specific workflow requirements, complex matrix analysis, or production environment operating constraints. We also have simple separations. These systems are ideal for those new to separation science. These core technologies, such as the Alliance HPLC or the BridgeQS HPLC, offer affordable, highly reliable, low complexity systems for routine analysis. And finally, purification systems. Proliferative scale HPLC plays a critical role in applications where compounds must be synthesized, identified, isolated, purified, and characterized. These systems enable substantial increases in throughput with an added dose of system robustness. But the chromatography separation itself is only one piece of the puzzle. We also need to collect, analyze, report, and secure our data to be able to make critical decisions with confidence. Our informatics solutions capture, manage, integrate, and analyze your LC and MS data, improving discovery and manufacturing, quality and integrity, and reducing time to market. Easily and seamlessly conduct scientific searches, integrate networks, manage compliance, and streamline operations. You invest significant capital in your LC system. Why not use the very best consumables? Waters is committed to developing, producing, and manufacturing innovative and consistent products for solving your most difficult challenges. As you can see here, our enabling technologies meet your standards for performance, reproducibility, and quality, giving you confidence in your analysis. As I mentioned before, Waters has added 2 new systems to our portfolio in the last 9 months, the Achilles PLC and the ACQUITY Premier. So lastly, let's take a look at the benefits of these products. The Achilles PLC was released in the summer of 2020. It is built on Waters' proven technology. Method transfer could be transferred between different labs or different vendor instruments or different separations platforms. The Arc HPLC ideally designed to show equivalent separations between different platforms, not only for Waters Instrument, but also other vendors' HPLC systems. We can use features such as Gradient Smart Start to facilitate this process, saving time, leading to more productivity and better asset utilization. If you want to improve your productivity and efficiency, this can be performed on an Arc HPLC as well because of its higher back pressure tolerance compared to the existing Waters HPLC. We can scale the method, moving to a 3.5 micron column, and all performance can be improved. And additional benefits include lower solvent purchase and disposal costs. As the next product, in February 2021, Waters redefined separation science once again with the introduction of the Acute Premier. This system solution integrates consumables, column chemistries, and the LC system to solve some of the most challenging separations in analytical chemistry with mass spec technology. What are the challenging separations today? Analyte loss is one of the most troublesome challenges in chromatography that labs experience today. This problem manifests itself in many ways: poor sensitivity, poor reproducibility, poor recovery, or even missing analytes. MaxPeak high-performance surfaces technology, which we are applying to the LC system, the column, and the consumables used in the Premier solution, are designed to improve sensitivity, increase analyte recovery, improve resolution, and minimize those interactions which lead to sample losses. Finally, as a new product, we are going to introduce another innovative new product based on this Premier solution platform. This new system is coming soon this year, and I'm very excited. This system will also bring innovation for your success again. Today, I gave you a brief overview of current Waters LC portfolio. Thank you very much for your time and attention. Please contact your Waters representative at any time if you have any chromatographic questions. Waters has a system solution to help you answer the questions with confidence. Thank you again for listening.
Thank you, Charlotte, for the great overview of our current LC product portfolio. Next, I would like to welcome the next speaker, Mr. Shanmukh Bhatti. Mr. Shanmukh Bhatti is a senior manager in the analytical department in Rallis, India. He has over 28 years of experience in analytical area with GLP, GMP, ISO platform. He is familiar with not only method development in HPLC, LC, MS, GC, GCMS, Multitop, but also has a wealth of experience for standardizing analytical method and improvised QC procedures as per ISO norms. He is widely active both in India and overseas in the small molecular field, focused on agricultural chemicals and pharmaceuticals. I will now hand over to Mr. Shanmukh Bhatti for his presentation.
Hello everybody, good morning. My name is Shanmukh K. Patil. I am heading the Analytical Department, Rich Rallis India Limited, R&D Bangalore. Let me thank Waters India Limited for giving me an opportunity to present pesticides perspective from a regulatory aspect. Coming to this pesticide understanding from regulatory perspective, I'll briefly cover about a general definitions of pesticides, formulations, and how it is being taken in a regulatory perspective. Few thoughts on that pesticide residues and what are the important factors in considering the pesticide residues, MRL, fixation, kind of regulatory terms. So let us move on to next slide. Here, pesticides broadly classified as technical and formulations. Technicals are the one which have more concentrated, like more than 90%, say. Formulations are the pesticide which are formulated using technical material. So as a definition, technical pesticide, a bulk pesticide manufactured in the concentrated form, that is purity and impurities are as per the registration requirement, because it has its own requirement to be called as technical. And as far as formulation is concerned, it is a stable combination of various ingredients designed to render the product useful and effective for the purpose claimed. The form of pesticide as purchased by the users. So the formulation based upon the desire, like various formulations are available, like WP, EEC. I will cover in the next few slides. So those are very stable combinations. That means the active ingredient present in the formulation should work as is required to be used. So that is formulation. And active ingredient is a term. It is a biologically active part of a pesticide present in the formulation. So this is the main element which controls the pests. So how the formulating technical pesticides helps? Because nowadays, like increasing population, we have been witnessing, we have to feed increasing population with the food grains which are to be sufficiently produced. This is the biggest challenge every country is facing. So unless we use a scientific usage of pesticides, we cannot feed complete population with the food. That is inevitable. The pesticide usage is inevitable, but judicial usage is the one which is challenging, because the crop production itself has a lot of constraints, among which major threat from pest which completely devastate the crop production, thereby it reduces the food production. So pesticides comes into picture, pesticides or pesticide formulations which scientifically control pests attack on the crops, thereby serving the mankind. So this formulation of a technical pesticide helps to optimize the amount of active ingredient at a target site, because directly technical, a pure technical cannot be used directly to the crop. So it is a diluted one, which is because the active ingredient to very small quantity is sufficient to kill the pest actually. So that has to be formulated in such a way that only targeted pests are affected, and this also helps in maintain the physical and chemical properties during storage, dilution and use. So once formulated pesticide, you should keep its vigor throughout the period of storage. That is the biggest challenge during the pesticide formulation. And enhance the biological performance. So formulation which we use, that has to enhance the biological performance, reduce the effects on the environment and non-target organisms. The target is to kill the pests. So non-target crops should not affect by usage of the formulation. Like when we spray the pesticide, it should kill only pests. It should not affect the crops or beneficiary organisms. And one more and most important aspect is that the product should be safe to handle, because usage of pesticides by large is the farmers. So farmers are not well educated to handle. I'm saying most of the farmers. So it should be, we have to make a formulation in such a way that it should be safe to handle, safety to mankind. So in formulating such pesticides or formulated product, having those advantages requires a lot of study and many factors which influence the formulation type. Not all formulation types are used for all pesticides or all crops. There is a difference between usage of one kind of formulation to other. So these factors influencing formulation types are physico-chemical properties of technical. So sometimes purity matters, like that physico-chemical properties like its density, is liquid, its powder, it is crystal nature, it is boiling point, melting point. So many factors are there which governs the formulation type. Then biological activity and mode of action, how exactly it affects, like surface or inner surface or some on the field or on the crop, on the air, like there's so many mode of actions also, it depends. Method of application, whether you are spraying, you are spreading, or granulation or mixing with the seeds, so the methods of application also determines. Safety and cost, of course, this is a very priority basis, we have to consider it. Then market preference, then comes regulatory requirements. So once everything is okay but regulatory requirement is not met, means it will not be marketed. So regulatory requirements are very stringent and we have to comply with that at all the time. So these are the conventional formulations. I have just listed here. Dust called DP or D, there is a code. That means the packet which you see the product, no, in that these are the codes mentioned: granules GR or G, soluble concentrate SL or WSC, soluble powder SP, emulsified concentrate EC, wettable powder WP or WDP, bait, and seed treatments also many kinds of the DS, LS, FS, WS. These are the things which are moving forward. Many new formulations are being water soluble granules, water dispersible granules, capsule suspension, CS. These are the new formulations, very new generation formulations which are very popular nowadays, and these are eco-friendly and water soluble. So many advantages to be say that content like CS, ZC and ZD, all these things, no? Those are require very meticulous formulation, and usage of the adjuvant, active ingredients are very calculative. Suspo emulsion, micro emulsion, oil dispersion. I'm not going to detail it everything, but just to have what are the formulation codes and new generation formation in the market. I just listed here. These are some more list: ultra low volume, tablet, emulsifier gel, aerosol, so on. So coming to the main aspect, like how we are living with the pesticide and how it is playing a role in helping mankind. So by and large, pesticides mainly used on crop to target or to kill the pest. Pest may be insect, pest may be disease. So pest is general term. So it will target on pest. However, the application of the pesticide on the crop or field or wherever it is essentially used, it will not go completely. There is a residue. There is a residue of pesticide present on the crop, on the soil, on the water, so on and so forth. So ultimately what happens after this age, the crop will be harvested and the grains are separated. That grains may be containing pesticides, water, soil, everything will be contaminated. Ultimately it will be a part of pesticide. There is a chance that man or the person, all who are using food, maybe intake the pesticides. So that's called residue. Pesticide residue by and large may be contaminated in the food material. So that residue, that the term is residue itself, we have to define like what residue and how much the residue. If the presence of pesticide itself is not a big thing, but how much residue is present in that is important. And what is the safety limit of residue and its management. Because when the constraint is that pesticide has to be used to survive, like I told you, I mentioned that to feed a increasing population we need to have a pesticide usage as inevitable. So in that sense, how can manage, how we can lower the residue level in the food, that is very important. So for that many regulatory things are coming. So those are the things which are how they are evaluating the toxicity of pesticides. Just I give an example how they go by the severity. If the LC50 is like, that means dose is less than 50 ppm, parts per million, a very highly toxic pesticide is categorized, avian dieteratis. Then 50 to 500 is highly toxic, 500 to 1000 ppm moderately toxic pesticide, to 5000 it is slightly toxic pesticide. If it is more than 5000 ppm residue, then it is practically non-toxic pesticide. Similarly, it varies with the species also. Then mammal is less than 10 ppm is very, very highly toxic. More than 2000 ppm is practically non-toxic. So based on the category, because each pesticide behave differently. Not all pesticides behave equally. So there are so many chemistries are involved to ascertain how the pesticides are being evaluated. So this is how the classification for the regulatory. And coming to the residues, what exactly residues means? These are the concentration of any specified substance in food, agricultural commodities or animal feed resulting from the use of a particular pesticide. Due to the usage of pesticide after the crop harvest, if there is a residue, that means the remaining remnant of that particular pesticide, it's called residue. So residue may not be only the parent compound. It includes parent compound, its metabolites, derivatives, reaction products, impurities. So mainly the highlighted part is toxicological significance. The pesticide residue which is present is highly toxic. The toxicological significance governs whether it is highly high toxic or severe or non-toxic. So here the parent compound will be there, in the residue metabolite will be there. Sometimes metabolite is very toxic, then the parent compound, vice versa also possible. Sometimes during the reaction, so different products are formed which are toxic again, but these products are formed due to this pesticide. So how this is how they related. And impurities from the, because impurities bound to happen to be there in the technical materials, that may cause lot of toxicology significance. So these are the how to ascertain the residue due to which and what, that has to be significantly calculated. There comes the term maximum residue limit. This is the regulatory term which is very much important in ascertaining whether it is your dosage is or formulation product is safe or not. So by and large, maximum residue limit or the maximum concentration of a pesticide residue, normally it is expressed in ppm or milligram per kg, that is legally permitted. That is important. because zero MRL cannot be possible. So legally permitted concentration where in or food commodities or animal feed, this has been set by codex or national regulatory authorities. So certain limits are being assigned. If you go to codex site or a lot of pesticide list with MRLs fixed with the particular crops, all the category wise you can see. So this is how they have fixed over the period of time based upon pesticide nature, based upon crop nature, based upon season. Various factors have been considered and fixed that okay, this is the MRL for this crop, like that. So maximum residue limit we should not exceed. So MRLs are derived from an assessment of residues found when crop is treated according to GAP. So not all whatever the field, some study trial has been taken and assessed for the residue, pesticide residue and coming to very high, that has to be considered. It is not like that. The good agricultural practice, under a good agricultural practice, if the crop has been raised and used as per the dose requirement pesticide, then analyzed. So based upon that, MRL will be fixed. Consideration of various dietary residue intake estimates and determination of comparison with the acceptable daily intake, this is called ADI, is another regulatory term, should indicate that food complying with MRLs are safe for human consumption. So ultimately human consumption happens for the food and materials. So those should be complying. That is called acceptable daily intake. If particular intake of that particular food is considered by continuously taking a food, if the pesticide residue is like 0.1 ppm, then doesn't have any effect, like that they will correlate it. So ADI and MRLs are very important terms to give the pesticide registration. So normally MRLs are legal trading limits. These are set by regulatory authorities on a conservative principle to ensure proper usage of the pesticide and as a precaution to reduce potentially harmful effects. So actually on the term like MRLs are not safety levels, because different countries have different trading levels based on agronomic needs, food habits, usages, climate, so many things. Many factors govern there, but generally do not have different safety standards. MRLs are not derived from ADI. ADI is one of term used for the human safety, but MRLs are not derived from ADI. MRLs are not toxicology. MRLs are not toxicological threshold concentrations at which if they are exceeded, toxic effects must automatically be expected. The theoretical. So coming to ADI, this is the limit of daily intake of a pesticide that can be ingested over the lifetime of an individual without appreciable health risk based on the facts known at the time, expressed in ppm, that is our milligram per kg body weight per day. This is how the ADI is expressed. These will be derived factors. So exposure of public to residues in food cannot exceed ADI. This is very important. Then TMDI, predicted maximum daily intake, theoretical, of pesticide residue based on assumptions of MRL levels of residues in the food and average daily food consumption per person. This is expressed in milligram of residue per person. Then PHI, that is post harvesting index, number of days which should lapse before harvest after the last treatment of the crops in the field with the pesticide. That means pesticide when sprayed, crop harvest has to be done, like say if the crop has to be harvested 14 days after last spray of the pesticide, then the amount will be residue will be less, like that. Pesticide application and harvest of the crop, very important to ascertain the MRLs. So actually speaking, mRNA ADI no direct relations based off the separate evaluation of a different data. So mRNAs are proposed only after establishment of the ADI. So here comes the importance of all analytical tools, because whenever we say residue, MRL, all these things are based upon the data generated by the analysis. So the analytical methods which to be rugged and validated, those methods which results are accepted. So then reliability of data, that is under which circumstances these data are generated, whether it is GLP, GAP, GFP, like that. So that matters actually. Then raw agricultural commodity also, which governs MRL, processing, how the sample processed, how the storage of the samples, and number of field trials. It's not one or two, so there is sequence. Dosages, agroclimatic zones also important, and which variety of crop is used for the particular establishment of MRL, and a part of commodity, like whether it is a grain part or stem part or root part. Those are matters for MRL clarifications. So as we see here, the limits very stringent. Earlier it was 0.1 ppm limit of detection, below which there is no instrument to detect. So as years increasing, I mean, goes on 60s, 70s, it is like 2000 ppb level we can see now. ppt level also we can, sub ppt level also we can analyze. So that is how the analytical tools or techniques are developed over the years. So MRLs are very important. So there will be all regulatory authorities continuously monitor the MRL levels of particular crop or pesticide based on fresh data, because climatic zones changes sometimes variety changes, so many factors are there. There's a continuous monitoring process. So fresh data is required, registered users and new users, and analytical methods. Earlier conventional methods are there to fix the residue level. Now we have advanced tools, so that now it will be affecting the MRL levels. So very stringent measures will be taken. Then fresh information on metabolism, toxicity, that also triggers to review the MRL. Storage stability of the compound in the commodities. So because pesticide productions with is not the same as old as 1960s or 70s, so many changes happens. So technical materials are produced in different sources, different reactions. So that also affects the storage stability of the particular pesticide, even though it is the same name. So that also triggers MRL review. Information on processing, this also. In India, so almost all registered pesticides have been established for the MRL. All new pesticides has to give, like no registration granted without MRL. MRL is a must. So even for all label expansion means once the product is registered for some crop, if you want to ensure the same product for different crops or different seasons, so label expansions, that also required MRL fixation. So how we fix MRLs? Information on chemistry of the compound, which we are going to give. Information on short and long term toxicity. Then NOAL, that's called no observable adverse effect level, and ADI, acceptable daily intake. This has to be established. Information on environmental behavior, metabolism including toxicity of metabolites, analytical methods which we adopt, what is the LOD, LOQ achieved, all the validation parameters considered, even for the metabolites, and details of recovery studies, details of field studies, location, to say so many factors which are important, number of treatments, seasons, other practices, multi-locational and multi-seasonal residue data. This is very important. everybody, even regulatory authorities, keep asking on this, and we have to supply this data. Trial design protocols for treatment of the sample, transport, storage, pre-harvest, interval, all data, even environmental data have to be submitted. Storage stability of residues in the samples. So many factors are there which are data required for the MRL. So when it comes to the validations of the parameters, so this just briefly how they classify as how analytical tools are being used. Normally most of the pesticides are volatile and even non-volatile also. So the classification based upon thermally unstable, thermally stable. High vapor pressure, go for the liquid chromatograph. Vapor pressure is a governing factor. Then this is a classification, general classification. GCs are there. GC and HPLCs are the most commonly used, under which again advanced technologies improving HPLC's performance, like UPLCs, and GCs are HRGC coupled with a mass spectrometer. So those are governing factors in establishing the MRL. So in our R&D we have a lot of we have HPLCs under Waters ARC HPLC, and we have LCM, SMS, COTQD, which are being used to assess the pesticide content in the matrices. They are more reliable, directed in such a way that it improves the confidence level on the analytical results delivered from these systems. So we are very much happy to use such systems which are giving us a confidence in delivered results. Those sensitivity and reproducibility are very important parameters in getting the accurate results. So those are complied by the ARC HPLCs. So coming to the registration in India, the overview. I am just giving overview. So identification, screening of the new product, the product being registered, then product will be selected, then it has to be added to the schedule, which comes under this one. Then we have to take a permit for import cases, and again we have to do the data generation, then collation of dose years, then online data submission is there. Nowadays all data are submitted online. Those data are screened by CIB and RC, reviewed by scientific experts. If they need any clarification, they will revert back with the query. Then we have to give the explanation on the queries. Then again submission of deficiency answers by the applicant. Then before that CIB, along with the data, we have to submit a sample. Sample of them is product. That product and analytical test report also. So parallelly they will also analyze the samples, that products, for its active ingredient. Then a review of CIB-RC happens. Committee will be formed. That final assessment of inclusion of that product, then approval, RC subject to fixation of MRL, then grant registration, manufacturing license will be given, then launch of the commercialization of the product. So in between, parallelly, we have to take the Pollution Control Board clearance and the production of the product, and FSSAI comes a big role here. MRL fixation, though FSSAI is the main functional regulatory body which is fixing the MRL. So this is how it goes in the process, because the data which we submitted for one product, so a lot of toxicology data plus crop production data has to be given. So it takes several years, sometimes more than 5 years to 15 years. Some products are registered even after 7 years, 10 years. So it depends on the pesticide what we are testing. So Now, briefly, so what we can say is we in rich R&D, these water systems are giving excellent confidence in releasing the results, just getting no QC query from regulatory authorities on submitted results. We are extremely happy to have AutoSearch PLC and LC measurements in our laboratory. Include residues are inevitable, but byproduct of pesticide use, the fact that they are found at all is only due to the significant advances in analytical chemistry, because analytical tool which specifies this is the residue of this particular pesticide, this is a particular pesticide metabolite, so that is very important. The tests are now so sensitive that detection level that can be reached is equivalent to detecting 1 teaspoon of salt in 1 million gallons of water. So such a sensitivity has been achieved here with the various companies, analytical tools. Levels even lower than that can be detected. The mere presence of a trace amount of a pesticide does not mean a product or water or food is unhealthy. So the toxic significance has been established, well established, with minimum level and maximum level and so many parameters that determines whether it is a safe or unsafe. So this is how the pesticide and pesticide residues are being highlighted in present scenario. I thank you once again for giving me opportunity to present on pesticide perspective on regulatory. I thank Waters India Limited. Thank one and all. Thank you.
Thank you, Mr. Shimokipati, for the informative talk regarding the regulatory requirement in pesticide development and manufacturing. So our next presentation is in the title of Expanding Detection Capability in SEC and APC. Unlike SEC ICP-MS and APC ICP-MS hibernation. This talk combines SEC and APC coupled with mass detection in order to do polymer containing heteroatom characterization. I am happy to introduce the speaker, Dr. Miroslav Yanko. Dr. Yanko is a senior research scientist and a polymer separation leader within the analytical science team of the core R&D organization at Dow Chemical. Currently, his research is focused on the separation and the characterization of polymers on both particle and molecular level by HDC and LC, including SEC, HPLC, LC-CAP modes using conventional and enhanced detections, as well as coupled and hibernated tactics. His latest contribution to the field of polymer separation and characterization is development of ultra-high pressure size exclusion chromatography, which is now commercialized as an advanced polymer chromatography system by Waters. I will now hand over to Dr. Yanko for his presentation.
Thank you for joining this talk. The topic of my presentation today is enhancing detection capabilities in size exclusion chromatography and advanced polymer chromatography, shortly SEC and APC, and more specifically I will cover online hyphenation of the size-based separation technique with inductively coupled plasma mass spectrometry, shortly ICP-MS. Before I start, I would like to acknowledge my colleagues Beta Snow and Patrick Freifogel for their contribution to this work. In the course of this presentation, after a short introduction of polymer heterogeneity and liquid chromatography modes, I will cover APC as a way to address the continuous demand for short analysis times, better resolution, and improved precision. I will also demonstrate the increase in obtained information about analyzed sample when multiple conventional detectors are employed. Next, I will present the example of both SEC ICP-MS and APC ICP-MS hyphenations and demonstrate that these are effective approaches to determine not only molecular weight distribution and dispersity of analyzed polymers, but also to determine the amount of heteroatom containing monomer and its distribution within molecular weight of analyzed polymers, thus increasing informational richness about analyzed samples. Let me start with the statement that only Mother Nature can make uniform polymers. Man-made polymers are dispersed materials distributed in more than one direction. In addition to a distribution in molecular weight, they show distribution in chemical composition and groups, tacticity, and molecular architecture or topology. To possess all these distribution with the single analytical technique is a very challenging task, if not an impossible one. It's clear that the simple analytical method cannot provide all the required information. Methods which do not involve any separation, such as osmometry, light scattering, viscometry, or even NMR spectroscopy, generally yield only an average of the property to be determined, such as molecular weight, chemical composition, and functionality. When information about the distribution of these properties is required, a separation step has to be included. Liquid chromatography and its different chromatography modes depicted in this image are often used. SEC and APC are chromatographic techniques that are most often used to determine molecular weight distribution, while HPLC and UPLC are most often used to determine chemical composition distribution. Liquid chromatography at critical adsorption point is used to possess N functionality and tacticity distributions, as well as molecular weight distribution of one block-in-block copolymers. To further enhance the richness of obtained information on analyzed samples, still within one chromatographic run, a coupling of two different chromatographic modes is required. We refer to that as a 2D-LC. This topic is out of the scope of this presentation, but is covered by one of my colleagues presenters in this section. In my presentation, I will adhere to single chromatographic mode, specifically SEC or APC, and the richness of the obtained information about analyzed sample will be achieved through the coupling of multiple conventional detectors or employment of enhanced detectors. The representative list of detectors used in liquid chromatography that I divided into two arbitrary groups labeled as conventional and advanced is presented on this slide. Enhancing the richness of information about analyzed sample can be easily achieved by combining several conventional detectors such as RI, UV, light scattering, and viscometric detector, by using detector from advanced group, or by proper combination of detectors from both groups, as it will be demonstrated in the course of this presentation. It has already been several years since SEC scientific community is taking advantage of APC technology. APC is application technique for size-based separation of polymers using columns packed with sub-3 micron rigid high pore volume hybrid particles combined with fully optimized low dispersion acuity LC system. APC is superior to SEC in speed, resolution, precision, and last but not least, sustainability. Typical SEC run can take from half hour to 1 hour, while APC analysis can be finished as short as in several minutes. In terms of resolution, I believe that presented figures speak for themselves, demonstrating much higher resolution power of the APC. In terms of precision, RSD below 1% can be achieved using APC technology, while RSD of 5 to 10% is generally acceptable for SEC. Let me just say that at early stages of APC development, with only UV and LSD detector option available, APC technique delivers only a limited amount of information about analyzed samples. However, today, the detection capabilities in APC are equal to those in SEC, and the information revealed about analyzed sample is significantly improved, as it is demonstrated on the next slide. The informational richness improves when we start to couple two or more conventional LC detectors together, as is depicted in these setups on the left, where APC is combined with the set of 4 detectors: RI, UV, light scattering, and viscometer. Delivered result is not only relative molecular weight, but also absolute one, plus information about hydrodynamic radius, radius of gyration, and the branching or conformational information. And in some instances, also chemical composition distribution within molecular weight, as is shown in those figures on the right. All that with the significantly shorter analysis times due to the speed delivered by APC technology. Although obtained information about analyzed sample is quite impressive, in some instances it still might not be sufficient. So in the next part of presentation, detector from enhanced group of detector, specifically ICP-MS, will be employed with attempt to further increase informational richness about analyzed samples. Let me also make clear that ICP-MS detection is feasible for substances that contain elements, I will refer to them as a heteroatom in the course of presentation, that are detectable by ICP-MS. These heteroatoms are highlighted in this table. There is a vast number of small molecules, monomers, and corresponding polymers and copolymers. Several of these structures are also shown on this slide that contain the heteroatoms detectable by ICP-MS. And there is also a great interest to know the fate of these heteroatom containing moieties when incorporated into polymer chain and in fully formulated products. When the information about the total amount of the heteroatom is only required, spectroscopic techniques such as XRF and ICP-MS alone are most often used. However, the result is only average value. So when the distribution of the corresponding heteroatom containing moiety is needed, a separation step has to be included. Our current state-of-the-art online SEC and APC ICP-MS setup is depicted in this slide. We utilize Waters Acuity APC separation module to achieve desired separations, and the key component is the ICP-MS instrument as an online detector. In this particular case, Agilent 7700 series ICP-MS instrument is shown and utilized as a very sensitive and highly selective online detector with a high linear dynamic range capable of detecting all elements from lithium to uranium, as it was demonstrated in the table on previous slide. So far, we have demonstrated successful utilization of ICP-MS as online detector in HDC, SEC, and HPLC separation modes, and most recently also in APC mode. In terms of compactable solvent, both volatile aqueous buffers and a wide range of organic solvents were confirmed to be compatible with ICP-MS detection. On this slide, let me briefly describe ICP-MS detection. Column effluent, actually our sample, is continuously pumped into nebulizer where it is converted into fine aerosol with argon gas. The fine droplets of the aerosol are separated from the larger one in the spray chamber. The fine aerosol is transported into the plasma torch. In plasma, at high temperature, atomization and ionization of the material take place. Subsequently, the ions are extracted into mass spectrometer, where the elemental composition of the material is determined. Now, I would like to present a couple of examples where combination of the resolving power and speed of size-based separation techniques, both SEC and APC, with the high selectivity and sensitivity of ICP-MS, made significant contribution to the discovery of unknown information that helped move customer project forward. First example is a separation and characterization of heteroatom containing polymer by SEC-RI ICP-MS hyphenation. Effluent from SEC column is split and monitored by refractive index, shown on this slide, and ICP-MS detector on the next slide. Using SEC charts of analyzed samples and the proper calibration curve, relative molecular weight data and fraction below 1,500 can be determined. While the first 5 lots of analyzed materials are consistent in terms of molecular weight, lot number 6, depicted here in cyan color, shows lower molecular weight and is identified as an outlier. SEC charts of the analyzed samples as detected by ICP-MS detector are shown here. There are 3 distinct peaks on the chromatogram. Peak number 1 is assigned as heteroatom response in the polymer. Peak number 2 is labeled as a residual or unincorporated heteroatom containing monomer, and peak number 3 is unintended heteroatom containing byproduct. Using calibration curve of heteroatom containing monomer on the upper right, the total amount and the distribution of the heteroatom containing monomer can be determined within polymer molecular weight and is summarized in table below. It's clear that twice as much of heteroatom containing monomer was charged in the sample 5. By combining information from both refractive index detector on previous slide and ICP-MS detector on this slide, 2 of 6 analyzed batches were identified as outliers. Next example is even more challenging one. There is a need to characterize heteroatom containing component in fully formulated system when component of interest, shown here in red trace, coalesces with other formulation components, shown here as a black trace, and its level in the formulation is low in the range of couple of percent as shown by the blue trace. In other words, this is an example when we are looking for a needle in the haystack. Shown here is an overlay of the SEC charts of the neat heteroatom containing component, blue chart, and the same heteroatom containing component in the fully formulated product, red chart, as detected by ICP-MS detector. Due to high sensitivity and selectivity of ICP-MS detector, a challenging task becomes quite a straightforward one. Broadening of molecular weight distribution of heteroatom containing component is revealed with the high sensitivity. It is hypothesized that the heteroatom containing component is undergoing chain scission and chain recombination during formulation process, resulting in the presence of low molecular weight and high molecular weight species. A summary of analysis of several lots of fully formulated products by SEC-RI ICP-MS hyphenation is shown on this slide. While no difference can be revealed by a refractive index detector, see insert in the top right corner, the ICP-MS detector reveals significant differences in the level summarized in the table on the left, and molecular weight and molecular weight distribution of the heteroatom containing components which correlated well with process variables. The disadvantage of the SEC ICP-MS approach is a long analysis time, usually 30 to 60 minutes, that become an issue when using solvents that char or carbonize a lot, causing a drift in ICP-MS detector signal. ICP-MS detector drift can be minimized and even completely eliminated by utilizing APC technology, which allows to achieve desired separation in the significantly shorter time. On this slide, 4 batches of heteroatom containing polymer were analyzed and compared using APC, with the analysis time as short as 12 minutes. Another advantage of using APC is that the range of the applicable solvent is greatly increased due to less strict column and liquid chromatographic system back pressure limitations. Due to short analysis time, the ICP-MS detector response stays linear over a wide range of concentration, as demonstrated by calibration curve on the left, and the amount and the distribution of the heteroatom containing monomer can be easily determined and compared. 3 batches from different vendors are quite consistent in the amount of heteroatom containing monomer, but 1 batch shows significantly lower amount. APC ICP-MS results were validated by independent technique, XRF, and good agreement between APC ICP-MS result and the results obtained by XRF is demonstrated. On this slide, I would like to address a potential concern of peak broadening caused by ICP-MS detector. Overlay of SEC charts of heteroatom containing standard covering broad range of molecular weights as detected by refractive index and ICP-MS detector is presented. Detector responses are high normalized and appear as a single chart, despite the fact that there are 2 lines. One smooth one is for a refractive index detector, and second, more spiky one is for the ICP-MS, suggesting no or minimal peak broadening caused by ICP-MS detector. With that, I would like to conclude that SEC and APC using conventional detectors, including RI, UV, MALS, and WIS, are very robust and cost-effective analytical tools, but provide somewhat limited information on analyzed samples. We have demonstrated that SEC and APC hyphenated to ICP-MS detector deliver significantly richer information on analyzed samples, but add to the complexity of experimental setup and data processing, resulting in longer turnaround times and higher cost. Further improvements in both instrument and software technologies are required to minimize the complexity of the experiment using advanced detectors, so that SEC and APC hyphenated technique become routine analytical approaches for polymer characterization in both academic and industrial settings. Last but not least, I would like to acknowledge the support from both Dow and Waters, and thank you for your attention.
Thank you very much, Dr. Yanko, for the great talk on hyphenated ICP-MS for SEC and the APC. Next, I would like to welcome Dr. Bastian Stoll to the next presentation in the title of Recycling Gradient in Polymer Chromatography, a new approach in HPLC on polymers. It will focus on interactive chromatography and how it could determine the chemical composition distribution of polymers. Dr. Stoll is a polymer chemist who finished his PhD in 2005 in the University of Eindhoven on copolymer characterization by multi-top MS. Since 2006, He is a lab leader at BASF, responsible for GPC and HPLC of polymers for the central research at BASF. His main interests including developing alternative methods in data processing for both HPLC and GPC, searching for stable conditions for performing proper GPC, unraveling multi-MS data of copolymers, and hibernated tactics such as GPC-IR, and pushing the boundary of 2D chromatography to its limit. I will now hand over to Dr. Stoll for his presentation.
Good morning or afternoon. My name is Bastiaan Staal, and today I would like to present you something about recycling gradients in polymer chromatography. Is this a new approach or is this just another curiosity? Before we're going to dive into all kind of details, I would like to take you on a journey from a perspective like what is it, why we are all doing this. And one of the main goals of characterization is understanding one of those fundamental relationships which we always like to picture. The chemical, the physical, and of course the application. And how do they interact with each other? Forget about the picture on the left. Unfortunately, things are more complicated if we want to understand the overall application properties. It is not just a linear combination of chemical and physical properties. But first of all, we need to characterize our samples. And with the characterization methods we use for polymers, we are basically close to the chemical properties which we can describe. Whether it's NMR or HPLC or GPC, if we want to know more about the physical properties, we have to use other techniques. Nevertheless, in many cases, understanding why an application is performance good or bad, it is simply defined by the difference between good and bad performance as samples. And that is the main task of characterization, to see if we can distinguish between good and bad performers. So why is it then so difficult? And why are we talking so much about the characterization? So what type of do we have then? Well, there are many types of different characterization techniques we can use for characterizing polymers. SEC is just one of them, or GPC, but there are many more. The problem is that there is not a single technique who gives the full picture of a polymer and characterizes it at once. So what is it then that it makes so hard to characterize the polymers? The problem with polymers is that they are never single distributions. They are distributions in distributions. We can think about the length of polymers. They can be long or short. They can be different in chemical compositions. That is already a challenge. And they can be different in typology, for instance, branched or not branched. And all those properties together form our final product or a final polymer with its characteristic properties. So as you can imagine, it is impossible to separate all those distributions simultaneously, and especially for industrial polymers, which normally consist of many more different components. Today, I will only focus on the characterization methods where we separate polymers, because only by separating we might have a chance to understand those underlying distributions. So what are then the standard methods for polymer separation? Because that's where we need to go to. Well, GPC, separation to size, is definitely one of them. Interactive chromatography is another very important. And there are many more, but those two are the main ones if you're talking about polymer separation. And all those methods require that polymers can be dissolved. So I will not talk about polymers in a solid state. But we need to dissolve polymers, otherwise, if they can be dissolved, all those methods cannot be applied. So that narrows down our methods which we can use to some extent. And one of them I already addressed, GPC or SAC, to determine the molar mass distribution, or actually it's the size which we measure. And the other one, interactive chromatography, is one of the systems where we can determine chemical composition or end groups or other kind of things. And today I would like to focus on the chemical composition distribution. So before we can continue, I need to make sure that you understand the principle of interactive liquid chromatography. I try to put a scheme here where green is the non-solvent and red is the solvent. Then at T0 we inject on top of our column our sample, which precipitates or absorbs there and doesn't move. And while the time increases, we move from a non-solvent to a solvent, then our peak starts to move and elute in a particular order until everything is out of the column. It is based on the fact that we are weakening the interaction with the column as we are moving towards a stronger solvent. In this sheet, we see an example of how separation according to chemical composition could work. In this case, we use a gradient from acetonitrile to THF, where we separate a copolymer SAN called styrene acrylonitrile. With the time, we increase the solvent composition from acetonitrile to more THF, and we see that peaks eluting from our system, which contains more acrylonitrile. This was measured offline as used as a calibration curve. That's why you see those 4 red peaks in there. The green sample was a commercial sample, which was used for comparison to see to what content this acrylonitrile was in there. So this shows the idea of how we can use interactive chromatography for separating according to chemical composition. But remember, this only works if there is no molar mass dependency. And to prove that is what we see at the next slide. By doing two-dimensional chromatography. If we combine our size exclusion method with our interactive chromatography method, we can actually see that this separation is not completely molar mass dependent. Especially in the low molar mass range, we see there is a strong molar mass dependency in the HPLC separation which we are doing. On the right, we still see the same picture as we see on the slide before. And remember, the part which is marked in red, where the green curve goes down, that is actually based to the molar mass dependency. You can also see that directly in the two-dimensional picture, because if the molar mass should be independent on the chemical composition, it should not have a banana shape, but it should become horizontal and not bended down. So that means if we go back to another example here for polyethylene glycols, which are lower molar mass, we see this effect even much more pronounced. What we see here is an HPLC separation, but from a homopolymer. On the left, we see the very low oligomers for polyethylene glycol, and the 3 ones to the right are standards with an increasing molar mass. So this is all nice and good because the separation looks very nice, but this is actually not what we want if we want to separate for chemical composition. We want to diminish this molar mass effect. Actually, we want to don't have any molar mass effect at all because it only jeopardizes our way how we have to look to our distribution. So congratulations to the people who made it so far, because now we finally can define our aim. Can we indeed diminish those molar mass effects of an HPLC separation for polymers? That is a very good question, and it took me a long while to think what we can do about it. So here comes the idea. If we use an infinitively long column in such a way that each polymer molecule have enough time to reach its equilibrium state within a gradient, we might reach our target that we do not have a molar mass dependency anymore. Wow, that's really a mouthful. So how does this work then in practice, if it works at all? Let's first have a look to the experimental setup. What we see here is that we use two columns, column 1 and column 2, like ordinary GPC columns, and they are switched in such a way that if we put a gradient in, we can recycle it. And I will show you it on the next slide how that works, because it's a bit difficult to read it from such kind of pictures like. So let's start at the beginning, where we are just doing an ordinary chromatography, in this case interactive liquid chromatography. On the left picture, we see column 1 is going to be filled with a gradient from our pump. Remember, green is the non-solvent and red is the solvent. Starting completely filled with ACN, and we see that the color to the right is red, is coming in our column, so our column 1 is going up in solvent composition. If you follow all those arrows, you will see that column 2 still remains green and is completely full with acetylnitrile. So that is what happens in the beginning at T0. Then we move to the right, and then we see something that at the end of a certain time, nearly all the gradient which I put in the first column at the beginning is simply flushed through the first column and parked or moved to the second column. You can also say, like, you simply moved the gradient from the first column with time to the second column. That is correct. And we notice that our pump has moved back from the gradient from a good solvent to our non-solvent, and very rapidly, and that's why we see this gradually changing of color very rapidly from red to green in our first column. So in this way, we have parked our gradient into the second column. Note that there is a UV detector between column 1 and column 2. If you follow the lines, you will find it out. But the summary here is, and that is very important, that we parked our gradient from column 1 into column 2. So, and now the fun can start, because what we are doing now is we are switching our valve from position A to position B. And what then actually happens is that the gradient, which is now in column 2, is starting to enter again the beginning of column 1. If we look to the picture on the right, we can see that again, after a certain time, the entire gradient from column 2 has moved back to column 1. And as you can imagine, this can be repeated all over and all over again. But before we can continue this, we have to put back our valve, of course, in position A, and then it starts where we were 2 slides ago. In this way, we can repeat our gradient, parking it from column 1 to column 2, and vice versa. Remember that the time which is needed is what we later will call the cycle time. And another aspect we should not forget is we need to move our gradient from, in this case, 100% A to 100% B within the column volume of the column we are using. So let's have a look at some results, because maybe it's a bit difficult to imagine. Every time you switch, it reminds me about a 2D chromatography. And that is actually how this data, the raw data, at least looks like. On the left, we see the UV detector, which was in between those columns, if you remember. And that just picks up, in this case, some signal of some polystyrene standards. On the horizontal axis, we see the time or the number of cycles, if you like. And every time there is such a black line, we are switching the valves from one position to the other. Down to the right, you see a magnified part where you can see the gradient, showing you how stable this gradient works. Another way of looking at those kind of data is to plot it in a 2D plot. And that might help to visualize actually if peaks are stable or not. If we are looking to this data set as a 2D data set, then it looks like this. And the first time when I saw those results, I got very disappointed. And the reason why I got very disappointed is because I was expecting that all those monomasses would migrate to a single point. Remember, I wanted to create a method which could measure molar mass independent. And even with those columns and very long runtimes, lower molar masses still seems to move, didn't become constant, and those higher molar masses never reach a single point. So besides the fact that those measurements took an ugly long time, I mean 250 minutes, that's by far too long for industry standards, it still did not merge to a single point. So I should came to the conclusion here that this method failed. So that triggers the question, what did I do wrong? Or what is it that I need to change? On the previous column, we were measuring on a silica column. That is probably the wrong type of chromatography for those kind of things. So we changed to a C18 column. We're using a bit of smaller dimensions, then the measurement would not take so long, and therefore we could do a gradient a bit faster in 3 minutes instead of 12 minutes. Again, down we see the same picture as what we saw before. But every black line is simply the switching valve moving from one position to the other position. Now we see here again our contour plot, or a two-dimensional chromatography plot. And one of the things that immediately should catch your attention is the low molar mass species to the left, the molar mass of 2,800 grams per mole. With each cycle, it is moving to the right. And indeed, after a certain number of cycles, all those individual standards here line up in a single line. And that is exactly what we wanted to achieve here, a separation where we can measure molar mass independent. I think some of you might get a bit confused and might wonder, like, I'm missing the whole picture. What was this all good for? So let's go back to our comparison, what we normally need to find those conditions. In liquid chromatography, we have a name for methods which we use if we measure molar mass independent, the so-called HPLC under critical conditions. And here we see an example how those critical conditions are found for an HPLC method where we are running isocritical conditions. That means, for instance, to the curve to the right, if we use 45% of acetylnitrile on this type of columns, we see that the PEC 600 elutes just before the PEC 4000, the red and the pink color. We also know if the higher molar mass elutes after the lower molar mass, we are clearly in the HPLC modus. If we then increase our content of acetylnitrile, so we are increasing our solvent strength because the non-solvent was air water, we are moving back into the GPC modus, and that is here what the blue course presents. By doing trial and error, you can find that for this particular column, that at 45.8% acetylnitrile, you have reached the critical conditions. Hooray, hooray. Indeed, PEC 600 and 4000 are eluting at exactly the same time. And the moment you achieve that, you can clearly say, okay, I found the critical conditions for my system at those temperature. As you can imagine, it is laborious to find this critical point. And now comes the fun part. By using this recycling gradient method, we end up exactly at the same critical conditions. The only thing what we need to do is recycle our gradient. So although we are still running a gradient, we will reach exactly the same critical point as what we do otherwise by trial and error offline. So this means that if I have to compare those methodologies, I know which one I will choose. The one to the right, because I don't need to look exactly for those conditions, because by cycling this gradient, it will find its critical conditions by itself. The only thing is, I don't know how long I need to cycle. And that brings my presentation to my conclusions. I've shown you today that recycling gradient is a potential method for reducing the molar mass dependency in HPLC separations. I've only shown it for a homopolymer, and I cannot show it for a copolymer, but that's something I will do in the future. Playing with a relatively simple setup, as I've been shown, it gains you a lot of insight in the separation mechanisms. We could clearly see that the same separations on a silica column did not work, and we can also clearly say that critical conditions on this silica column, however, how bad you tried or whatever you did, they simply do not exist. And this mechanism can also indicate you that you will not find them because they don't exist. So this means if it proves that this works indeed correctly or in the way we think, then we do not only have find an easier method for screening if critical conditions exist, but we also find other potentials how to combine this type of gradient, of recycling gradient, for also future 2D LC setups.
Thank you very much, Dr. Stoll, for the informative and the unique approach addressing 2D chromatography. The last topic for today is presented by my colleague Sato-san on silane coupling agent reaction monitoring with APC. Nobutake Sato-san has over 10 years experience specializing in mass spectrometry and the separation tactics for the characterization of our materials. He currently is the Asia Pacific Material Science Marketing Manager for Waters Corporation and oversee how Waters product adopted in the area of polymer, fine and specialty chemicals, extractable and leachable, medical device testing, cosmetic and personal care products, agrochemicals, and fuel and energies. Over to you, Sato-san.
Good morning, good afternoon. I'm Sato, Materials Science Marketing, Waters Asia Pacific. Today, I will briefly introduce an application of silane coupling agent reaction monitoring using APC. This is the agenda for today. Before talking about the application, I will introduce what is silane coupling agent and what is the analytical challenge we have. Silane coupling agent is a compound whose molecule contains two functional groups that can bond with organic and inorganic materials, respectively. If we look at the structure, functional group X can bond with organic materials, and functional group on the right can bond with inorganic materials. Silane coupling agent acts as a sort of intermediary which can bond organic materials to inorganic materials. By bonding inorganic and organic materials, various properties are added to inorganic materials, such as heat resistance, durability, water resistance, and mechanical strength. For this reason, this is widely used in industry, such as for tires, inks, building materials, films, and electronic parts. However, the silane coupling agents are generally known as highly reactive compound. This alkoxy group reacts with the water to form silanol group. This silanol group is very unstable and can undergo condensation reaction over time. When not reacting with materials, this results in formation of siloxane linkage and ultimately gelation. For this reason, analytical technique which can measure changes in the reaction of reactant and byproduct is very important. Next is analytical challenge. Generally, as I mentioned, silane coupling agent has high reactivity with water and it's very unstable compound. So there is no proper technique that can analyze coupling agent at stable state. For example, like NMR, this is non-destructive analytical technique and can perform quantitative and qualitative measurement, but it takes time to obtain enough intensity. In case of GC, gas chromatography, this cannot directly measure monomers and oligomers, and in many cases, derivatization is required. GPC, this is one of the options to avoid water, but very poor separation capability and takes longer runtime. HPLC, in many cases, water is used for mobile phase. This is an example of reverse phase LC. The chromatogram on the top is the total ion chromatogram. We can see two peaks here, but these two are not reactant-related peaks. These two are degraded component of reactant. If we look at the chromatogram on the bottom, this is an extracted ion chromatogram of coupling agent. As water is used for mobile phase, immediately coupling agent reacts with water and degraded. Therefore, we cannot see any response on the chromatogram. For the measurement, we need to minimize the reactant to be converted to the silanol state during analysis. One of the keys is using high-speed and high-resolution GPC. GPC, side exclusion chromatography, usually does not require water for mobile phase and provide less interaction with current. Another aspect is using same alcohol as a functional group in reactant for mobile phase. By using same alcohol, we can keep the reactant in its initial structure, which means we can suppress the reactant becoming silanol. This is APC, Advanced Polymer Chromatography System we use for this time. APC is high speed and high resolution size-based separation chromatography. So faster speed and higher resolution compared to conventional GPC system could offer some advantages for reaction monitoring. This time we tested two silane coupling agents. One is tetraethoxysilicate, known as TEOS, TEOS. The other one is acrylic-based silane coupling agent. The time when we mixed up coupling agent, solvent, and catalyst in a vial is reaction start time, which means time 0. The reaction stop is the time when this solution is injected into the system At the moment, catalyst becomes diffused or kept away from the reactant. The first example is TEOS. I don't go through the details of analytical condition, but one remarkable is to use ethanol for reaction solution and mobile phase, which is the same alcohol as functional group of reactant. This is RI chromatogram at time 0, which means before reaction starts. The peak around 8 minutes is peak of TEOS. This is an overlay RI chromatogram from 1 minute to 24 hours after the reaction. If we look at the TEOS peak here, as time goes by, peak height of TEOS decreases. On the other hand, we could see the peak eluting before TEOS. This peak may be a dimer and oligomer and increase over time. If we use a conventional ZPC system, these components cannot be separated by size exclusion mode. One of the purposes of reaction monitoring is to determine some chemical kinetic factors, such as reaction order and half-life. These factors are very important to understand what types of reactant it is and what the reaction depends on. For TEOS case, half-life is 104 minutes, and reaction order is first-order reaction. So first-order reaction means the reaction is dependent on only the concentration of reactant, and reaction is not dependent on reaction time. The second example is acrylic-based silane. This is analytical condition. This time, we also use the same alcohol as reactant. In this case, we use the methanol. And we use UV detector and mass detector for the deeper elucidation by mass number. This is UV chromatogram before reaction, which is time 0. Only the peak corresponding to the reactant was detected around 4 minutes in elution time. This figure shows the time course of reaction. The chromatogram on the left is a total ion chromatogram of time 0, 1 minute, and 120 minutes after the reaction. And on the right is the mass spectrum. So if we look at the spectrum at time 0, the most abundant spectrum is methoxy dissolved precursor ion of reactant. And also we could see protonated precursor ion here. 1 minute after the reaction, we could see another peak appearing before the reactant. Looking at the spectrum of this component, this may be a precursor ion of dimer according to the mass number. Then, 120 minutes after the reaction, another peak appeared here, which may be more than trimer, and reactant peak had completely gone. This is an extracted ion chromatogram of protonated precursor ion of reactant. The highest intensity peak is time 0, and peak intensity decreased over time. As we can see this chromatogram, the time when the peak height becomes half is about 30 minutes. Then we calculated chemical kinetic factors. The half-life of this coupling agent is 29 minutes, and reaction order is zero order reaction. So zero order reaction means reaction rate doesn't depend on initial concentration of reactant. So even if we change the initial concentration of reactant, that doesn't provide any effect on the reaction speed. The reaction speed is just dependent on reaction time. Therefore, when we obtain these chemical kinetic factors, we can leverage these factors as indicators in order to develop the product with well-controlled reaction rate, or to determine the concentration at formulation, and as well as to consider usage conditions. This is my last slide. Styrene coupling agent is a highly reactive compound, and there were few analytical techniques. This time, we adopted high-speed and high-resolution GPC system, and this could achieve reaction monitoring of styrene coupling agent. Based on the acquired data, since we can calculate the chemical kinetic factors, we may be able to accelerate new material development through the control of its reactivity. That's all from me. Thank you for listening. And if you have anything on this, please contact us. Contact Walter. Thank you for listening today.
Thank you, Sato-san. We have now come to the end of our session. Today, we have heard from a strong line of speakers on different analytical strategies for materials innovation. In pesticide development and manufacturing, they require proper analysis as per regulation. The sensitivity and the reproducibility that influenced by chromatography accuracy are very important parameters in getting accurate results. In the talk provided by Dr. Yanko from Dow Chemical, it shows that the advanced SEC technology could maximize the effect of a hibernation with selective detectors to support complex polymeric materials characterization. Also, Dr. Stoll from BASF shared a unique approach that applied to DLC to help to understand the characterization of complex polymer materials. The high-speed and high-resolution SEC technology can also enable us to monitor the reaction of a high-reactivity compound that could not be achieved in the past, and this brings the first-to-market advantage. Lastly, as a reminder, you will be able to access today's recording after this live event, and we will consolidate all questions and answers by our speakers as a post-event email follow-up. Thank you very much for your attention, and we at Waters hope you have found the material science section informative and enjoyable. Have a good rest of the day.